Good morning, my dear students. Today we shall take up a topic, HMP shunt pathway. This is in the pathway of the carbohydrate metabolism and carbohydrate metabolism. In the carbohydrate metabolism, one of the important aspect is HMP shunt pathway. Shunt pathway, shunting. That is where it is started, it is come to again back to the same place, going all around and coming back to the same place, shunting. So, what is this shunting and thing? Let us see. This is an alternative pathway to glycolysis and DCA cycle, Krebs cycle. This is an alternative pathway to glycolysis and DCA cycle. Krebs cycle for oxidation of glucose. The glucose, we know that it is the primary source for all the animal beings, the source of energy. Once the glucose molecule is broken down oxidatively, the first part is called as glycolysis. The next continuity in the Krebs cycle are tricarboxylic acid cycle, and the last here, certain soluble substances which are called as whose name is NADH and FADH2. These two soluble electron equivalents. These compounds will be entering into the next respiratory chain of events, and ultimately energy is liberated. This way, once glucose is completely gets oxidized, finally into carbon molecule, that is in the carbon dioxide form, and the hydrogen ions in the water form, they will come out and then produces a lot of energy. And that energy, almost 36 moles of energy from one energy means ATP molecules are generated from 1 mole of glucose. This is for the sake of energy production. For other purposes, suppose two molecules to join together in the body and produces another new molecule. This is what is called as anabolism. The one which we have discussed, that is producing energy, is a catabolic process. Glucose getting catabolized by oxidation, producing energy. Similarly, the fatty acids also gets oxidized and produces energy. That is fatty acid oxidation. So oxidative process, a degradative process, catabolic process produces energy. But from smaller molecules joining together, producing the larger molecules of by importance in the cell are all called as anabolic activities. In the anabolic activities, to join two compounds, you need energy. And this energy is in the form of NADPH. These are also called as reducing equivalents. NADH and NADPHs are called reducing equivalents. And these equivalents, one is producing energy, the another one is producing the energy required for joining of the molecules. And anabolic nature, this HMP pathway is anabolic in nature of producing NADPHs. Since it is concerned with the biosynthesis or otherwise production of NADPH and another important substance from the glucose, these are the pentoses. Why we need pentoses? Why we need the pentoses? What is the expansion of DNA? Deoxyribose. Ribose is a pentose. And then RNA, ribose nucleic acid, deoxyribose nucleic acid. So both the nucleic acids needs the 5 carbon sugars. And hence the synthesis of DNA, that means the synthesis of nucleus, when is it needed? When the cells are getting divided and then producing newer cells. So when the cells are producing their daughter cells and progressing the multiplication in the process of cellular multiplication, pentoses are needed. So for these two substances are coming in this alternating pathway, that is the HMP pathway. Unique multifunctional pathway. This is a unique multifunctional pathway. And the enzymes located in cytosol, whereas oxidation of glucose, first part glycolysis is in the cytosol, and TCA cycle and Krebs cycle is in the mitochondria, and the respiratory chain events in the mitochondria. So two compartments, whereas this is completely in the cellular compartment. All the enzymes are present in the cellular compartments. And the most important is the production of NADPH. So what is the main aim of the HMP pathway? This is a oral question. This is the production of NADPH and the production of pentoses. Again, production of NADPHs and the production of pentoses. What for NADPHs? Reductive biosynthesis and reductive power in various reactions that are occurring in the cell at various levels. And then pentoses for nucleic acid synthesis. So now, whenever there are reductive biosynthetic processes are occurring, and there the HMP pathway is active. So, in which tissues the HMP pathway is very active? One is liver. What does it do? Liver detoxicates various xenobiotics, various foreign substances entering into the body are all being detoxicated in the liver cells. And hence, for those reactions, we need a reductive power in NADPH. Adipose tissue, fatty acid synthesis. Synthesis of fatty acids requires NADPH again. And hence, this tissue have the HMP pathway active. Adrenal gland. This produces steroidal compounds, corticosteroids, and then sex steroids. And these steroids are the mother molecule is cholesterol. The cholesterol is totally synthesized de novo in the cells from acetyl-CoA. You may supply from the extraneous source via nutrition, but they can produce completely the cholesterol, various tissues, and one active tissue, adrenal gland. Another active are testes and ovaries. Testes and ovaries. These gonads, they are all active. Adrenal gland and gonads are active in the synthesis of these hormones. And hence, they are all the mother molecule cholesterol. Cholesterol need to be synthesized from the acetyl-CoA in toto, and for that purpose, NADPH is needed. And hence, here the HMP pathway is active. Erythrocytes. The most important is erythrocytes. NADPH is very, very, very essential, because in the erythrocytes, the nascent oxygen insides the membranes and produces what is called as peroxidative attack. Peroxidative attack, or otherwise oxidative attack. This oxidative attack has to be nullified, neutralized by a substance called as glutathione, which is present richly in erythrocytes. This glutathione, to prevent this oxidation, it gets oxidized. And once it gets oxidized, it can no more functions. It needs to be brought back to reduced form. For that purpose, the NADPH is needed. And hence, HMP shunt pathway need to be active in the erythrocytes as well. And another one is lactating mammary gland. What is the lactating mammary gland? Needs two important things. One is fats need to be synthesized to mix in the milk. So milk is rich in fats, triglycerides. And then another one is lactose. The synthesis of lactose, the synthesis of fatty acids, all are needing again the reductive power NADPH. And hence, these tissues are actively having this shunt pathway. That is the reason. what are the other names for this HMP, hexose monophosphate shunt pathway? Phosphogluconate pathway, because the next substance that is being produced from the glucose 6-phosphate, and basing on that, this is the phosphogluconate pathway. As here, the pentoses are produced, and again they are rearranged to produce glucose 6-phosphate if they are not needed. Then, and hence it is a cyclical pathway touching the pentose and going back to glucose 6-phosphate. And hexose monophosphate pathway or shunt pathway as the classic name. And the scientists who are involved mainly in this deciphering or discovering this particular cycle of these events, Warburg, Dickens, and Lippmann pathway. These are the great Nobel scientists who has completely deciphered the HMP shunt pathway. Here, from the glucose, various pathways are there. Actually, from the glucose 6-phosphate is produced. So the central pivotal point, central main junction is glucose 6-phosphate. From the glucose 6-phosphate, one can produce the storage form of glucose, that is glycogen. This is one side. And when it is needed, glycogenolysis and get glucose 6-phosphate. This is one side. And another side, from the glucose 6-phosphate may be passing through the oxidative pathway, which is glycolysis, producing pyruvate, and from the pyruvate it can again diverge either into anaerobic path producing lactate or aerobic path going into the TCA, going into the finally respiratory chain, and finally producing carbon dioxide and water and energy. This is one way. And another way is glucose 6-phosphate, and it can, well, in the pathway reversal of glycogenolysis, it may produce glucose. This is by glucose 6-phosphatase, and it may be circulating in the blood. Apart from all these things, another path from glucose 6-phosphate is pentose phosphate pathway, in which the most important things produced, ribose, that is the pentoses, and NADPH. And let us see in details the HMP shunt pathway from the glucose 6-phosphate, or by some other way glucose 6-phosphate might come. And then now the first step in the HMP shunt pathway, is by the participation of glucose 6-phosphate dehydrogenase enzyme, which is very, very, very important. This is popularly called by the physicians G6PD, deficiency hemolytic anemia. The medicine persons very much fond of this particular diagnosis. Glucose 6-phosphate dehydrogenase G6PD deficiency. This is occurring in a lot of genetic populations, various populations. We'll discuss later. And what is the participation here? Coenzyme participation in this enzyme? Glucose 6-phosphate dehydrogenase acting on glucose 6-phosphate and taking the help of NADP, nicotinamide adenine dinucleotide phosphate is the coenzyme, and magnesium divalent cation also participates here and produces the NADPH and H+. So the reduced NADPH is being produced. This is essential. What is the substance produced? 6-phosphoglucanolactone. 6-phosphoglucanolactone. And now, with the participation of a molecule of water, which is not depicted here, participation of water, glucanolactone hydrolase, then here the 6-phosphogluconate is synthesized, is produced. And now another reductive enzyme, phosphoglucanolactone, another dehydrogenase. So two dehydrogenases and two levels NADP is converted to NADPHs, NADPHs, and which are reduced forms. And where from this NADP? Produced from a vitamin. Which is that vitamin? Can you say? Niacin. What is this niacin? Is the component of B complex. What is the name of it? B3. What is the deficiency? What is the deficiency? Deficiency of niacin, pellagra. So, okay. So now the coenzyme from niacin, NADP, is being reduced to produce reductive molecule NADPH. And now what is being produced is ribulose 5-phosphate. Ribulose, a keto group, and 5-carbon sugar, pentose. So it is a keto pentose and 5-phosphate. Ribulose 5-phosphate is produced. And what is the peculiarity or specialty of this particular enzyme is a carbon dioxide is produced out. Normally, carbon dioxide comes only after the respiration is complete. Respiration is completely complete, and oxygen will be taken and then producing with the help of the skeleton of carbon, and carbon dioxide is coming in the respiratory chain only. But this is a speciality in the HMP shunt pathway. Carbon dioxide is liberated in the cytosol, nowhere else. Nowhere else. So there are two enzymes. This is also called as oxidative phase of HMP shunt. Oxidative phase of HMP shunt. Now, next part. Let us see. This ribulose 5-phosphate. Imagine one molecule of ribulose 5-phosphate. Now, it will be acted upon by two different kinds of enzymes. One is isomerase. When you call it isomerase, a ketose will be converted to an aldose, or an aldose will be converted to a ketone. It is a bidirectional. Though it is not written, it is a reversible reaction. A ribose 5-phosphate is an aldose. The ribulose 5-phosphate is a ketose. A ketose is producing an aldose, and aldose is producing a ketose. Then you call it isomerase. Several times I have told you in the classes. Isomerase. So one ribulose molecule is acted upon by isomerase and producing ribose 5-phosphate. Simultaneously, another ribulose 5-phosphate molecule is acted upon by epimerase and producing. Now this is enomerism. So one carbon, a hydroxyl group on one carbon moiety shifted to another carbon moiety, and that is what is called as enomerism. Enomerism. A group is producing, shifting from one point to another point on different carbons of the same molecule. This is what is called as the epimerase. So another ribulose molecule 5-phosphate. Ribulose 5-phosphate molecule acted upon by epimerase and producing xylulose 5-phosphate. So you have one aldose and one ketose, and acted upon by one is epimerase, another is isomerase. And let us see. Now this aldose and this ketone having 5 carbons. From this ketose group, along with the keto group and the above carbon both, so 2 carbon piece from the xylulose 5-phosphate is cut by an enzyme called as transketolase. 2 carbon molecules are being cut from xylulose along with the keto group and is being transferred onto the head of the ribose 5-phosphate. Already this is a 5-carbon sugar, and 2 more carbons has come, and along with a keto group has come, and hence now 7-carbon ketose sugar whose name is pseudoheptulose, and as the phosphate now in the end 5 instead now 7, so this becomes in the end 7-phosphate. pseudo-heptulose 7-phosphate. The remaining 3 carbon part along with its last phosphate. Now it is having only 3 carbons now. So now this is glyceraldehyde 3-phosphate. Hope you are understood. So the enzyme here trans-ketolase. And here the coenzyme form which is being present participating in this reaction TPP, thiamine pyrophosphate. Thiamine pyrophosphate. Thiamine pyrophosphate is synthesized from or active form of which vitamin? Thiamine. Thiamine is again a member of B complex group. What is its number? What is its location in the B complex? The first position, thiamine B1. So thiamine pyrophosphate is a coenzyme in trans-ketolase, and also you all know in other enzymes, other important enzymes, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase. In those again branch chain keto acid dehydrogenase. In these 4 enzymes, thiamine pyrophosphate is a coenzyme form. But the enzyme which participates in the HMP pathway is trans-ketolase. What is it producing? It is making use of 2 5-carbon sugars. One is an aldose, another is a ketose, and then producing a 7-carbon keto sugar and a 3-carbon aldose sugar. So theme is almost a aldose, ketose, but a piece is being transferred onto this, and then one became 7, another is 3. Total number of carbons are 10 again. Here 10. But there is a rearrangement, molecular rearrangement. Now, next comes. Next, what will happen? Again, another enzyme now called trans-aldolase. Trans-aldolase. This trans-ketolase transacts 2-carbon compound, whereas trans-aldolase shifts, changes 3-carbon compound. Where from we will get the 3-carbon compound? Now, from here. From here. What shall we do? From 3 carbons, we will cut from here. Along with the keto group. Along with the keto group, the top 3 carbons, we will cut from the 7 carbon. So, from the 7, 4 carbons, 3 carbons are cut along with keto sugar, ketose moiety. Then the remaining is 4 carbon, and as ketose is lost, then remaining one is an aldose only. So erythrose 4-phosphate from here. And that 3 carbon along with the ketone, that is the moiety, whole moiety is dihydroxy acetone moiety is being fixed on the top of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate and dihydroxy acetone, both of them are producing the fructose, and as the 3-phosphate becomes now the 6-carbon molecule, and in the last it is there, and hence automatically it becomes 6-phosphate. So a fructose 6-phosphate is generated. Where from we have started? We have started from glucose 6-phosphate. So from fructose 6-phosphate, you can have glucose 6-phosphate without any difficulty, as the enzyme is reversible. Is a reversible enzyme, isomerase enzyme. So now, that is, the ketose can be altered to an aldose. Similarly, here an isomerase can have, and then glucose 6-phosphate you can have if no reaction takes place further. So now, other side, erythrose 4-phosphate. That means the 6-carbon became 4-carbon. 6-carbon, you got it, but 2 molecules of 6-carbon, they lost as carbon dioxides, and now you have a 4-carbon moiety here. Now, imagine this erythrose 4-phosphate. Now here, erythrose 4-phosphate. And also imagine xylulose 5-phosphate, 1 more molecule. So that means 3 molecules have come in the pathway, and ribulose 5-phosphates 3 molecules. 1 molecule goes into the isomerase path, 2 molecules goes in the epimerase path, and 2 molecules of xylulose. 1 xylulose has gone in this pathway, another xylulose 5-phosphate here. You got it. and now this one and this one again acted upon by the same enzyme transketolase. So transketolase, transaldolase, and transketolase again. What can it do? It cuts two carbon compound along with the ketone. Along with ketone. Where is ketose present? Ketose present is here. So from the xylulose, the 2 carbon moiety is cut. Remaining is going to be 3 carbon compound. What is that 3 carbon compound? Glyceraldehyde 3. And the ketose group and 2 carbons are fixed onto the erythrose 4-phosphate. 4 plus 2 is 6 and is having a keto group and hence fructose 6-phosphate. So here one fructose 6-phosphate, here one fructose 6-phosphate, and finally glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So 3 molecules of glyceraldehyde 3 participated, 3 molecules of ribulose biphosphate, all the 3 molecules passing through all these things and producing ultimately fructose 6-phosphate this side, fructose 6-phosphate this side, and glyceraldehyde 3-phosphate. So that means 3 carbons have been lost from 3 molecules of the glucose 6-phosphate, and this is ultimately a glucose 6-phosphate. This is ultimately a glucose 6-phosphate, and this is half glucose 6-phosphate, you can say. So 2.5 molecules, 3 molecules finally producing 2.5 molecules of glucose 6-phosphate. And now imagine another 3. Similarly, another 2 molecules of glucose 6-phosphate and half molecules. And so 2 glyceraldehyde 3-phosphates mixed together and producing another fructose 6-phosphate and glyceraldehyde 3-phosphate. So 6 molecules of glucose 6-phosphate after running into these 6 cycles and producing 5 molecules of glucose 6-phosphate. So 1 molecule is vanished. You see? So you need to understand in this way on multiple cycles of a HMP pathway. Then only you can understand. So now this is being shown. So in chemical rearrangements and other things are being depicted. Glucose 6-phosphate, glucose 6-phosphate dehydrogenase. NADP participation, NADPH you have participation. And after losing the hydrogens, here is a hydrogen. And then what has happened? Here you see 6-phosphoglucanolactone. 6-phosphoglucanolactone, OH. You see? And this H is lost. The O has come here. And the rearrangement is happening here. 1 H, 2 H lost, and the H are here. So NADPH. Next is water is entering into it. 6-phosphoglucanolactone hydrolase, and this you see, then this became 6-phosphogluconate, and then 6-phosphogluconate dehydrogenase. Again another NADP participation, and NADPH is being produced, and a carbon dioxide gone out. Where from carbon dioxide? The last. The acidic moiety of COOH in the 6-phosphogluconate has been comes out as carbon dioxide. Ribulose 5-phosphate. Ribulose 5-phosphate by isomerization producing ribose 5-phosphate, by the epimerization produces xylulose 5-phosphate. And I said this is been cut here and then is been added to this and producing here pseudoheptose 7-phosphate, and then the becoming losing it, this becomes erythrose 4-phosphate. Erythrose 4-phosphate, pseudoheptose 7-phosphate. No, glyceraldehyde. This side is going. This is glyceraldehyde 3-phosphate. And now this glyceraldehyde 3-phosphate. Erythrose, no. Erythrose. And then pseudoheptose 7-phosphate is this side, and this side is glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphate. This trans-ketose after trans-aldolase. In the trans-aldolase pathway, this glyceraldehyde 3-phosphate is being depicted here. There is a little confusion here, please. So, it is depicted here. And now, these two acting upon by trans-aldolase and producing 3 molecules are cut. 3 molecules are cut and added on to the glyceraldehyde 3-phosphate, producing fructose 6-phosphate, and then the remaining one erythrose 4-phosphate. And then that erythrose 4-phosphate and then erythrose 4-phosphate and another xylulose 5-phosphate acted upon by another transketolase, producing fructose 6-phosphate and glyceraldehyde 3-phosphate. So previously one fructose 6-phosphate, here is another fructose 6-phosphate with the phosphohectose isomerase, it producing glucose 6-phosphate. Similarly, there also glucose 6-phosphate and 2 molecules of glucose 6-phosphate, half molecule of that is glyceraldehyde 3-phosphate. So another 3 more cycle and 2 molecules of glucose 6-phosphate and half molecule of glucose 6-phosphate, that is here 1 molecule. So 5 glucose 6-phosphates will be coming out. So the whole thing is called as non-oxidative phase. Oxidative phase, non-oxidative phase. So it is been divided into oxidative and non-oxidative phases. This is oxidative phase and this is all non-oxidative phase. What is the significance of HMP pathway? No, this is more important than the molecular rearrangements. Pentoses or its derivatives are useful for the synthesis of nucleic acids and nucleotides. NADPH is required for reductive biosynthesis of fatty acids and steroids, number 1. For the synthesis of certain amino acids, reductive power. Antioxidant action, very important. And the hydroxylation reactions are derived in the liver for the detoxification of drugs. And the most recent advances of phagocytosis in the neutrophils needs the NADPH. Preserve the integrity of RBC membrane. Oxidative attack on RBC membrane is prevented. Antioxidant reaction, what is this? Glucose upon hexokinase producing G6-phosphate and G6-phosphate producing NADPHs. And NADPHs are very, very essential to reduce the oxidized glutathione. Oxidized glutathione is reduced back and this becomes potent reductive power. When potent reductive power is there, oxidant materials here and there will be nullified by reduced glutathione. Reduced glutathione acts as antioxidant, most important. You see, in the pentose phosphate pathway, the hydrogen ions are added on to the oxidized NADP and producing reduced NADP. Now, there is an enzyme called as glutathione reductase. This uses FAD as a coenzyme. And when the oxidized glutathione is available in the cell, with the help of glutathione reductase and NADPH, which is liberated in the pentose phosphate pathway, using FAD is a transacting mediator, now producing the glutathione oxidized to glutathione reduced. This is very, very important. And oxidized NADP. This oxidized NADP, again one more cycle of pentose phosphate pathway passing through, becomes reduced. Now, this reduced one, what is the next action? The reduced one is used for glutathione peroxidase enzyme. Glutathione peroxidase enzyme is having the selenium as the associate. And glutathione peroxidase converts the H2O2 to H2 and water, and nascent oxygen. Nascent oxygen. So now, glucose 6-phosphate dehydrogenase deficiency leads to inherited sex-linked trait. It is a inherited sex-linked trait. G6PD deficiency genetically present in various populations, especially around the Mediterranean population, around the Mediterranean Sea population. And few people in the North India, Punjab people, and there is a deficiency. Red blood cells are going to be, there is a deficiency, red blood cells are going to be broken down fast, and a hemolytic anemia. Impaired synthesis of NADPH. This is the deficiency occurs, impaired synthesis of NADPH. Hemolysis and developing hemolytic anemia. Resistance towards malaria is one of the advantages why the nature has selected the mutation of glucose 6-phosphate dehydrogenase. Especially this is in the Africans. The clinical correlation is G6PD deficiency results in hemolytic anemia, neonatal jaundice, and kidney failure. Again, G6PD deficiency results in hemolytic anemia, neonatal jaundice, and kidney failure. And in the RBC, the activity is being again shown. Glucose entered into the RBC, glucose 6-phosphate, like a stack. And G6-phosphate, it can go in the anaerobic glycolysis producing lactate and producing ATP. This is a normal way. But this glucose 6-phosphate can go in an alternating pathway, and in the glucose 6-phosphate dehydrogenase producing NADP to convert the NADPHs are liberated. Okay, the activity is going in the forward direction, leave it, but our liberated NADPH, what is going to happen? What is it going? The glutathione reduced is oxidized glutathione. Oxidized glutathione is converted to reduced glutathione. This reduced glutathione, the reduced glutathione is acted upon by the glutathione peroxidase. Along with glutathione peroxidase, the H2O2 present in the cells, how they are obtained, certain drugs, infections, and flavins also produces this oxidant material, H2O2. And this in the respiratory chain, after production of energy, you are also getting H2O2. And anyway, this glutathione peroxidase and reduced glutathione, they acted upon the H2O2 and then producing the water molecules, 2 H2O molecules. And again, producing oxidized glutathione. Oxidized glutathione again becoming reduced. This cycle is important, otherwise here it fixes, here it stops, and then H2O2 is increased, build up, and then this is oxidant attack on the membrane lipids, and peroxidation attack, and then the membranes are broken down, red blood cell is broken down and producing hemolytic anemia. Metabolic significance of HMP shunt. It is the only source of phosphorylated pentoses which used for the synthesis of, what are the synthesis of nucleotides, ATP and GTP, and they may produce nucleic acids. It also produces coenzymes, FMN, FAD, NAD. What is that? Phosphorylated pentoses. That is ribose biphosphate, glucose biphosphate, etc. They are going to participate in the synthesis of these substances. Certain coenzyme forms of certain vitamins. Synthesis also needs these. That is B2 and B12 needs to be pentose phosphates. And ultimately, nucleic acid synthesis, DNA and RNA. And the deficiency of this thiamine pyrophosphate, you know all, the genetic disorder associated with HMP shunt, an alteration in the trans-ketolase activity in the HMP pathway, trans-ketolase activity altered, and it reduces the affinity with the thiamine pyrophosphate and the biochemical lesion of Wernicke-Korsakoff syndrome. What is this? Korsakoff syndrome characterized by amnesia, forgetfulness, creation of new memories, and apathy. not feeling like interesting in anything, lack of interest in every activity. So forgetfulness, lack of interest in any activity, and new memories, creation, without not having really, but a new creation occurs. So this is Korsakoff syndrome. Both of these diseases, Wernicke and Korsakoff, both are caused by B1 deficiency. Simultaneous occurrence makes Wernicke Korsakoff combined syndrome. Another one is fava beans. Fava beans. Fava beans are consumed by especially these Mediterranean people. They are something like our what is this? Chana or various pulses we eat and consumes, and fava beans are consumed by them, like maize and groundnuts, something like these ones. Here, fava beans are consumed by those people, Mediterranean people, Greece, and then Italy, etc. Those people, and even Egypt people. And these people, when they eat these beans in large quantities, when they have the glucose 6-phosphate deficiency is there already, then more quantity of H2O2 is liberated by them oxidatively, and then this H2O2 cannot be nullified because of lack of NADPH production, and excessive H2O2 liberated, and membranes of the RBC are broken, and hemolysis. So immediately after eating a large quantity of fava beans in these population, they are going to have hemolytic crisis. So that is the danger of eating fava beans there. This child, a child with favism, the signs of anemia, decreased red blood cell count, and jaundice occurs because a lot of anemia occurred. So hemolytic jaundice occurs also. So I have seen so many cases because I had the privilege of working there in Saudi Arabia as director of laboratories, and hence we have seen a lot of number of cases. So this is how it's saying. So this is about the HMP pathway. Ultimately, what is the message? HMP pathway is an alternative pathway. It starts from glucose 6-phosphate and gives again glucose 6-phosphate. And in the pathway, you have two important, very, very important substances produced. And what are they? One is NADPH, reductive power. Another one is the pentose phosphates. What is the significance of pentose phosphates? Just now we have learned that it produces various nucleotides and various vitamins and various coenzymes, and ultimately various nucleic acids, that is DNA and RNA. See, this is one thing. And another one, reductive power. Reductive power is antioxidant. And the detoxification mechanism, it will be assisting, and it is assisting in the phagocytosis. It is maintaining the RBC membrane integrity. If it is not, there is going to be hemolytic anemia. The frequent commonest disease, one of the commonest cause for hemolytic anemia, G6PD deficiency, especially in the Mediterranean people and those populations where G6PD is prevalent, like Punjab of India. So now, this is finally about HMP pathway and the important substances produced, NADPH and the pentose phosphates. So this is what the HMP pathway uses and the applications. Thank you very much.